Assalamu alaikum dear students, it is nice to see you again on this video and this lesson. As previous lessons, we will follow these objectives in this video and you will be able to understand these objectives. First, we're going to talk about cellular respiration, then step by step we will talk about glycolysis, about the Krebs cycle, and also about the link reaction, and we will end up today's lesson or this video by oxidative phosphorylation. In order to talk about cellular respiration, we end up with a result that cellular respiration causes the ATP production. And now we know that ATP is called adenosine triphosphate, which is the energy currency that is going to be used by living cells. And so where ATP is going to be produced, we also know about the energy storage, that they are main fuels which are going to be used inside cellular respiration. These main fuels are the result of photosynthesis. It means the carbohydrates, lipids, and proteins that we eat inside our food and drinks, they are going to be exchanged from their form into usable energy currency that we call it ATP. So in this equation you see that this is glucose and this is oxygen. Together they will combine in order to produce carbon dioxide, the water, and energy, bypassing different type reactions of oxidation and reaction, in which hydrogen is going to be passed to attach with the oxygen to produce the water, and also the carbon is going to be attached with oxygen to produce carbon dioxide, and at the end it will result for production of the energy which enables the living organisms in order to carry out different living functions. Let's talk about the steps of the cellular respiration. The ATP production step begins with glycolysis. To understand glycolysis, we can say that this is a reaction in which glucose is going to be split and converted into pyruvic acid. At the beginning of this red row you can see that is a glucose, a hexose, which possesses 6 carbons, and it will end up with 2 molecules of pyruvate that possess 3 carbons. Also it is called triose. So in order to have another step in cellular respiration, it is really necessary that glucose should be split into different parts. In order to split the glucose and convert it into a usable shape of energy that should enter in next step of cellular respiration, we should take these 2 steps. First, we should understand about phosphorylation, that inside glucoses or glycolysis, phosphorylation happens. It means glucose is going to be converted into different and other sugars, for example fructose phosphate, fructose biphosphate, then it is going to be converted or split into 2 molecules of triose. And here, as you see, to carry out the phosphorylation function, 2 ATP is needed to add in this reaction. So at the end, when glucose splits into 2 molecules of triose phosphate, you can see that ATPs are produced. Also NADs or electron carriers are also produced. So at the end of glycolysis, we can summarize that for starting the glycolysis we spent 2 ATPs, but at the end we received 4 ATPs and 2 reduced NADs. It means the electron carriers which accepted the electrons, and carries out into further step of cellular respiration. About the link reaction, we can say that this is the second step of cellular respiration. Link reaction means when the pyruvate passes by active transport into mitochondrial matrix, and there it will face these 3 steps. First, it is decarboxylated. It means from these 3 carbons, one of the carbon is going to be split and going to be exhaled outside of the cell in the form of carbon dioxide, and also the hydrogen which is attached with the NAD or electron carriers are going to be forwarded into next steps of cellular respiration. The second is dehydrogenated. As I mentioned, that NADs will receive the hydrogens. The third step is this, that In link reaction, pyruvate is going to be converted into acetyl-CoA or acetyl coenzyme A. When we enter inside the Krebs cycle, we will face with different circles and different enzymes and reactions. But in order to summarize the function of Krebs cycle, we can say that we should follow the 3 steps in order to understand the Krebs cycle function. Mainly, to define the Krebs cycle, we can say that the Krebs cycle is a closed pathway of enzyme control reaction. It means inside the circle, there are different enzymes that controls the exchange or transform of different molecule of sugars into ATP. At the first step, we can see that acetyl-CoA here is going to be combined with oxaloacetate here. The result is citrate here. Acetyl-CoA is a kind of molecule that possesses only 2 carbons. Oxaloacetate is another kind of sugar that possesses 4 carbons, and 2 and 4 combines together to make a citrate that possesses 6 carbons. So furthermore, in another step or second step, this is the citrate, which is going to be dehydrogenated and decarboxylated, and the result will be the carbon dioxide, and also the electron carriers are going to be produced in the name of NAD and FAD. So the third step here, as you see, oxaloacetate is going to be regenerated in order to carry out this continuous circle of the Krebs cycle. So when we reach at a final stage of cellular respiration, we will should be familiar that oxidative phosphorylation will happen inside the inner membrane of the mitochondria. In cell organelles, we studied that organelles are called little organ of the cell, and one of those organs in eukaryotic cell were mitochondria. Mitochondria could be thought as a powerhouse of different cells of animals and plants. So oxidative phosphorylation will happen or take place in the inner mitochondrial membrane. In order to understand the exact functioning units of the oxidative phosphorylation, we should go in these steps. First, the reduced NAD and FAD. When we talk about reduced, it means we are talking about reduced electron carriers that they have attached the hydrogens with them. So NAD and FAD are passed to the electron transport chain. This is called the electron transport chain. The second step, you should be familiar with these 4 respiratory complexes, which are embedded in the inner membrane of mitochondria. So their function is this: to exchange electron one by one. At the third step, when the protein pass to enzyme, channel ATP is synthesized. As you see here, this is called ATP synthase. It means this is the enzyme which uses the energy of electron, which is carried out to these respiratory complexes and caused that to these protein channel different concentration gradient of the hydrogen ion should be created outside of the inner membrane between the outer and inner membrane of the mitochondria. Then the energy which is going to be used by this electron, by using that energy, ATP synthase will use the potential energy of the hydrogen ion in order to produce the ATP, and at the fourth step the cell will be able to use the ATP or adenosine triphosphate as an energy currency to trade between another type of cells or carry out their livelihood functions. But at the end of the cellular respiration, we will end up with this result, that ATP production will start with the glycolysis, then it will enter another step in the link reaction, the Krebs cycle, and it will end up with oxidative phosphorylation. The total income and consumed energy and the cellular respiration could be like this, that ATP used in glycolysis were 2 ATP and ATP made in glycolysis were 4 ATP. So the net gain in ATP is 2 ATPs in glycolysis. And link reactions, we didn't spend any kind of energy. And Krebs cycle, we will produce 2 extra ATPs, but in oxidative phosphorylation we will end up with 28 extra ATPs. That the result will be 32 ATPs. That will enable the cell in order to carry out their functioning units. Thank you so much, dear students. That was your today's lesson. See you on next video.